look in the eye. You're just like an angel. Your skin makes me cry. You float like a feather in a beautiful world. I wish I was special. You're so very special. But I'm a creep, and I'm a widow. What the hell am I doing here? I don't belong here. I don't care if it hurts. I wanna have control. I want a perfect body. I want a perfect soul. I want you to notice when I'm not around. You're so very special. I wish I was special. But I'm a creep. I'm a widow. What the hell am I doing here? I don't belong here. Whatever makes you happy, whatever you want. You're so very special. I wish I was special, but I'm a creep. I'm a widow. What the hell am I doing here? I don't belong here. I don't belong here. You and me, we used to be together, every day together, always. I really feel I'm losing my best friend. I can't believe this could be the end. It looks as though you're letting go, and if it's real, well, I don't want to know. Don't speak, I know just what you're saying, so please stop explaining. Don't tell me 'cause it hurts. Don't speak, I know just what you're thinking, and I don't need your reasons. Don't tell me 'cause it hurts. Our memories, they can be inviting, but some are altogether mine, frightening. And as we die, reasons. Don't tell me 'cause it hurts. It's all ending. I gotta stop pretending who we are, who we are. You and me, I can see you sighing. Are we? Don't speak, I know just what you're saying. So please stop explaining. Don't tell me 'cause it hurts. Don't speak, I know just what you're thinking. And I don't need your reasons. Don't tell me 'cause it hurts. Don't speak. Every breath you take, every move you make, every bound you break, every step you take, I'll be watching you. Every single day, every word you say, every game you play, every night you stay, I'll be watching you. Oh, can you see you belong to me? How my poor heart aches with every step you take, every move you make, every bow you break, every smile you fake, every claim you stake, I've been watching you. Since you've gone, I've been lost without a trace. I dream at night, I can only see your face. I look around, but it's you I can't replace. I feel so cold and I long for your embrace. I keep crying, baby, baby, please. Oh, can't you see you belong to me? How my poor heart aches with every step you take, every move you make, every bow you break, every smile you fake, every claim you stake, I've been watching you. Every smile you fake, every claim you stake, I've been watching you. Every smile you fake, every claim you stake, I've been watching you. Every smile you fake, every claim you stake, I've been watching you. Every smile you fake, every claim you stake, I've been watching you. I used to think maybe you love me, now baby I'm sure. And I just can't wait till the day when you knock on my door. Now every time I go to the mailbox, gotta hold myself down. Yeah, 'cause I just can't wait till you write me you're coming around. I'm walking in sunshine. Oh, I'm walking in sunshine. Oh, I'm walking in sunshine. Oh, and don't it feel good? Hey, don't it feel good? I used to think maybe you love me, now I know that it's true. And I don't want to spend my life just in waiting for you. Now I don't want you back for the weekend, not back for a day. No, no, no. I said, baby, I just want you back, and I want you to stay. Yeah. I'm walking in sunshine. Oh, I'm walking in sunshine. Oh, I'm walking in sunshine. Oh, and don't it feel good? Don't it feel good? Yeah. Oh, don't it feel good? I'm walking in sunshine. Oh, I'm walking in sunshine. Oh, I'm walking in sunshine. Oh, and don't it feel good? I feel the love, feel the love, feel the love that's really real. I feel the love, I feel the love that's really real. And I'll say it again, oh yeah. I say it again, yeah, yeah. I'm walking on sunshine, baby, yeah. What the world needs now is love, sweet love. It's the only thing that there's just too little of. What the world needs now is love,
Yo no sé cuándo hay que... Se abre la sesión. Tiene la palabra don Francisco de la Torre, alcalde de Málaga.
Señora, honra su presencia en la ciudad de Málaga y honra siempre a los españoles su sensibilidad e implicación en temas tan importantes para la sociedad como el que protagoniza el tercer Congreso Internacional de Investigación, Innovación y las Enfermedades Neurodegenerativas. Quiero saludar también estas primeras palabras a la señora secretaria de Estado de Investigación e Innovación, y sabe que tiene siempre mi apoyo en su importante tarea en la Administración española. Saludo también al señor consejero de Salud de la Junta de Andalucía, al señor director del Instituto de Salud, Carlos III, y, como no, al delegado del Gobierno en Andalucía, a la señora consejera de Educación, presidente de la Diputación, señor rector de la Universidad de Málaga, a los organizadores de este importante congreso, este importante encuentro científico, y a los señoras y señores congresistas. Sean bienvenidos a esta ciudad hospitalaria, mediterránea, ciudad natal de Picasso, ciudad cuya estrategia se basa en la cultura, la innovación, el respeto medioambiental, la accesibilidad y la inclusión social. Han preparado ustedes, el comité organizador lo ha hecho con gran cariño y con gran esmero, este tercer congreso de CIIEN, el 11º Simposio Internacional sobre Enfermedades de Alzheimer y el 9º Foro Científico Cibernet. Saludo a los magníficos ponentes que han venido desde Potomac, estado de Maryland, de Estados Unidos, desde Londres, de Cambridge, de Madrid y Bilbao, Barcelona, Sevilla, Lisboa, Estocolmo, Santander y San Sebastián, y también de Málaga. A todos ellos y a todos los asistentes, mil gracias por trabajar para superar uno de los desafíos mayores que tiene la humanidad en el siglo XXI y que resumiríamos en una población que, con una esperanza de vida creciente, produce una situación en la que cada 3 segundos se diagnostica un nuevo caso de Alzheimer en el mundo, y con unas pirámides de población, con una base cada año más reducida, va a hacer que sea muy difícil sostener el estado de bienestar. Por otra parte, está el lado humano de estas enfermedades neurodegenerativas, el sufrimiento de los afectados y de los familiares, y también la pérdida que supone para el mundo dejar de contar antes de tiempo con la experiencia, la sabiduría, la capacidad de trabajo, de consejo y guía de tantas personas válidas. Por eso, vuestro trabajo en detección precoz mediante biomarcadores, vuestro trabajo en buenas prácticas de salud, terapia adecuada, e incluso los trabajos sobre predisposición genética y posible actuación en esta línea de trabajo son de una enorme importancia. Permítame que les comente, dentro de las terapias y de la prevención, cómo la ciudad de Málaga viene desarrollando desde hace 17 años un proyecto de memoria y demencias que se traduce en que en este año son 35 los talleres de memoria dirigidos a personas mayores de 60 años, que se reparte por el conjunto de los distritos municipales y asociaciones de mayores. Casi 1000 mayores se benefician de estos talleres. El Programa Municipal de Memoria y Demencias cuenta con un amplio reconocimiento profesional en nuestro entorno, habiendo recibido diferentes premios y distinciones en congresos y reuniones científicas andaluzas y nacionales. Asimismo, el Centro de Investigaciones Saludables ha sido premiado como la mejor iniciativa pública en el campo del envejecimiento activo, premiado en 2013. Gracias, en definitiva, por todo lo que trabajáis en este sentido, y espero que la reunión de Málaga sea muy útil para compartir experiencias y avances en vuestros trabajos, para plantear nuevas estrategias y programas de colaboración y compromisos de inversión pública y privada en vuestras investigaciones. En este día, 21 de septiembre, dedicado a recordar en el mundo la importancia de la enfermedad de Alzheimer, esta reunión de Málaga es sin duda uno de los actos más importantes que tiene lugar en todo el planeta, yo me atrevo a decirlo así, en relación al recuerdo y al compromiso para resolver este auténtico desafío. Gracias a todos de nuevo, mucha suerte y mucho ánimo.
Tiene la palabra don Jesús Fernández Crespo, director del Instituto de Salud Carlos III.
Buenos días, Majestad, señor delegado del Gobierno, señor alcalde de la ciudad de Málaga, señora secretaria de Estado, autoridades, ponentes, asistentes, organizadores. Es para mí un honor, en nombre del Instituto de Salud Carlos III, participar en este acto inaugural del tercer Congreso Internacional de Investigación e Innovación en Enfermedades Neurodegenerativas, que además engloba el 11º Simposio Internacional sobre Avances en la Enfermedad de Alzheimer y el 9º Fórum Científico de Cibernet, organizado por la Fundación Reina Sofía, el Ministerio de Economía y Competitividad a través del Instituto de Salud Carlos III y la Fundación CIIEN y Cibernet. En primer lugar, quisiera agradecer el esfuerzo, la dedicación, el compromiso y la sensibilidad de Su Majestad la Reina Sofía, así como de la fundación que lleva su nombre, hacia este gran problema social que es la enfermedad de Alzheimer y que ha hecho posible, con el apoyo de instituciones estatales, autonómicas y locales, que el complejo proyecto Alzheimer sea un hecho y esté funcionando en estos momentos a pleno rendimiento. En ese complejo proyecto Alzheimer participa el Instituto de Salud Carlos III a través de la Fundación CIIEN, que gestiona su unidad de investigación y el Cibernet. Igualmente, quisiera sinceramente dar la enhorabuena a los organizadores y coordinadores de este simposio internacional por el programa, por los excelentes conferenciantes que durante estos días nos ilustrarán con los hallazgos más recientes y relevantes en el campo de las enfermedades neurodegenerativas. Me gustaría destacar, en primer lugar, que la Fundación Centro de Investigación en Enfermedades Neurodegenerativas, Fundación CIIEN, está financiada por el Ministerio de Economía y Competitividad y adscrita al Instituto de Salud Carlos III. En el año 2011, y a propuesta del Instituto de Salud Carlos III, la Unidad de Investigación Proyecto Alzheimer y el Cibernet fueron designados Centros de Excelencia en Investigación sobre Enfermedades Neurodegenerativas por la Unión Europea. Esta, la Unidad de Investigación Proyecto Alzheimer y Cibernet, son las dos únicas instituciones en España que están participando en el programa conjunto para enfermedades neurodegenerativas, el Joint Program Neurodegenerative Diseases. La Unidad de Investigación Proyecto Alzheimer dispone de excelentes infraestructuras, adelantos técnicos y tecnológicos, junto con una masa crítica investigadora de Cibernet que han permitido a ambos organismos integrarse en la Red Internacional de Centros de Excelencia en Investigación sobre Neurodegeneración, COEN, una iniciativa surgida a partir del Programa Conjunto de la Unión Europea para la Investigación en Enfermedades Neurodegenerativas. Este programa conjunto es una iniciativa innovadora de investigación colaborativa creada para afrontar los crecientes desafíos que plantean este grupo de enfermedades y cuyo objetivo es potenciar el impacto de la investigación mediante la alineación de programas nacionales de investigación existentes y la identificación de objetivos comunes y la generación de una cultura y unos valores comunes. En 2014, algunos de los grupos de investigación de Cibernet han participado activamente en proyectos internacionales, como los 3 proyectos Pathfinder de la Red Internacional de Centros de Excelencia en Neurodegeneración o los 4 proyectos dentro de las convocatorias del Programa Conjunto de la Unión Europea en este área de investigación JPND. También merece la pena destacar algunos de los premios otorgados a lo largo del año 2014 a los investigadores, como el Paper of the Year Award, concedido por la American Neurological Association al doctor Miquel Vila, o el Movement Disorder Research Award, que la prestigiosa American Academy of Neurology otorgó al doctor Eduardo Tolosa como reconocimiento a su investigación centrada en la fase de la enfermedad llamada premotora de Parkinson y su contribución significativa al conocimiento, tratamiento y prevención de los trastornos del movimiento. En enero de 2014 se constituyó la plataforma Red Nacional de Biobancos, promovida por el Instituto de Salud Carlos III, de la que, por supuesto, forma parte el Banco de Tejidos de la Fundación CIIEN, y en el año 2014, para que tengan una idea cuantitativa, se inscribieron 106 nuevos donantes al Banco de Tejidos de la Fundación CIIEN, que cerró el año con más de 600 personas inscritas. Además, el Banco de Tejidos de la Fundación CIIEN renovó el pasado año su certificación de calidad conforme a la norma ISO 9001-2008. Durante el año 2014, el Departamento de Neuroimagen también ha realizado más de 5.700 estudios de resonancia magnética a 687 sujetos y desde la creación de este departamento se han realizado más de 38.000 secuencias. Finalmente, quiero sinceramente desearles que este congreso sea muy fructífero, muy provechoso y que les facilite seguir activos en este área específica de investigación con el fin de mantener la esperanza en que los resultados de los proyectos en marcha, así como de los que vengan en el futuro, proporcionen soluciones a este tipo de enfermedades que aumenten a mejorar tanto su calidad como calidad de vida para los pacientes y para los familiares. Muchas gracias por su atención.
Tiene la palabra doña Carmen Vela, secretaria de Estado de Investigación, Desarrollo e Innovación.
Majestad, señor delegado del Gobierno, señor alcalde de la ciudad de Málaga, consejero de Salud, señora consejera querida del Ávila, de Educación de la Junta de Andalucía, señor director del Carlos III, queridas amigas, queridos amigos, si me permiten ustedes, empezaré agradeciendo, es como se debe de empezar, y agradeciendo de nuevo, pero no por ello menos sincero, la presencia de Su Majestad la Reina doña Sofía, que con su permanente compromiso con las enfermedades degenerativas en general y con el Alzheimer en particular, no es más que un estímulo para todos nosotros para seguir trabajando en la búsqueda de soluciones nada fáciles y, por tanto, más esforzadas. Dicho esto, quiero, en nombre de la investigación y la innovación de nuestro país, dar la bienvenida a los numerosos investigadores y expertos que durante estos 2 días, os vais a reunir en esta triple reunión, en este triple simposium y congreso. Para Málaga, si me permite, señor alcalde, y para España es un verdadero placer y un verdadero honor contar con todos ustedes. Tienen en estos días por delante la oportunidad de escuchar, de debatir, de compartir y de intentar buscar soluciones para uno de los desafíos más importantes a los que nos enfrentamos, la salud. Pero quizá, en concreto y hoy aquí, la salud del envejecimiento, la salud de todas esas enfermedades asociadas con el aumento de la edad. Un desafío global, un desafío enorme al que no solo España, sino Europa se enfrentan y no de manera accidental han sido considerados, tanto en la estrategia española como en el programa Horizonte 2020, el reto número 1. El aumento de la expectativa de vida, debido efectivamente al aumento y al progreso del nivel económico y del nivel social, y créanme, y estoy segura que ustedes lo comparten, y un éxito para la investigación y para la innovación, hemos conseguido en estos países a lo largo de este siglo. Sin embargo, nos queda mucho por hacer. Y el tema del envejecimiento es, sin duda, un desafío científico, un desafío de salud pública, derivado del beneficioso, pero como con estas consecuencias, progresivo aumento de la edad y, por tanto, con estas enfermedades, tanto neurodegenerativas como cardiovasculares, asociadas a este problema. En este contexto, la demencia es hoy una de las condiciones degenerativas observadas con más frecuencia en las personas de edad y dentro de estas, Alzheimer es la forma más común de la dolencia, con hasta la fecha, y quiero insistir, hasta la fecha, todavía tratamientos no muy eficaces y con importantes consecuencias no solo para el enfermo, sino para el entorno familiar y, por tanto, sociales y económicas. Somos plenamente conscientes de las implicaciones que tienen estas enfermedades, de la necesidad de potenciar los medios, de potenciar las capacidades para todas, para hacer un abordaje completo desde la generación del conocimiento, desde la búsqueda de los mecanismos moleculares, desde el conocimiento de qué es lo que está pasando, para después orientar ese conocimiento y buscar mejores métodos tanto de diagnóstico como de terapia. Los tiempos, ustedes saben, los recientes, no han sido fáciles. No han sido fáciles, a pesar de lo cual esta Secretaría de Estado y este Ministerio hemos apoyado al máximo de nuestras capacidades a la Fundación CIIIEN a través del Instituto de Salud Carlos III. Se han seguido manteniendo las convocatorias anuales, las redes temáticas, tanto RETIC como CIBER. Queremos seguir en esta apuesta, queremos aumentar nuestros recursos porque sabemos que hay un enorme problema por resolver. Y sabemos, además, que nuestro colectivo investigador, con los recursos de los que dispone, los utiliza eficientemente. Prueba de ello son las 600 publicaciones con índices de impacto superiores a 5 que han mantenido en el año 2014. Es nuestra voluntad que sigamos trabajando juntos, porque el reto es de tal envergadura que solo juntos lo podremos abordar. Los científicos, las empresas, los médicos, los pacientes, los familiares, las administraciones, todos puestos en la misma dirección, trabajando juntos para resolver un problema y no solos. No solos porque un problema global requiere apoyo internacional y por eso, según decía el director, estamos en proyectos internacionales, por eso les quiero reconocer y agradecer la participación en el reto de salud de todos ustedes en Horizonte 2020, aumentando, y no es trivial porque la competencia en Europa es dura, el retorno para España en biomedicina de un 5,5% en el 7º programa marco a un 9% en las recientes convocatorias de Horizonte 2020. Y eso se debe a dos cosas. La primera, que ustedes participan. La segunda, que ustedes son capaces de competir con cualquiera. Y no hay nada mejor que competir con cualquiera y poner calidad y excelencia para resolver el problema que hoy tenemos entre manos. Termino. Majestad, autoridades, estimados colegas, amigas y amigos, en esta responsabilidad que asumo con un enorme ganas de trabajar, de estar con ustedes, de seguir en la misma línea en los momentos buenos y malos, resumo como empecé, agradeciéndoles a ustedes que estén hoy aquí, pero sobre todo agradeciéndoles el trabajo que cada día ponen en los laboratorios, en las oficinas, en el sitio donde desarrollen su trabajo, porque este reto, este problema que hoy nos concilia es enorme y solamente hay una manera de abordarlo, juntos y con mucho trabajo y con mucho esfuerzo, el que ustedes ponen cada día. Muchísimas gracias.
Se levanta la sesión. Muchas gracias.
Majestad, autoridades, colegas y compañeros, welcome to this Congress. This meeting will be mainly focused on different aspects related to dementia and movement disorders. And among the participants, we will have several foreign invited speakers. And in addition to our College of CIIIEN and from other Spanish institutions. The relevant foreign speakers are, in the slide, Professor Kachaturian from USA, Professor Rubinsztein from UK, Professor Rossor from UK, Professor Sedaso Minguez from Sweden, and Professor Costa, that is not here, but he will arrive in several minutes, from Portugal. Thank you, all of you, for coming here and for staying with us. After that, because we are a little late already, we are going to start with the first conference. The first lecture will be presented by Professor Zaven Kachaturian, and I know that Professor Kachaturian received his academic training in places like Yale, Cleveland, or Columbia universities. and is one of the main pioneers studying how to prevent Alzheimer's disease. Indeed, he is now the president of the campaign to prevent Alzheimer's disease by 2020. Professor Kachaturian is widely recognized as the architect of several successful international programs in Alzheimer's disease along his career. He served as the dual role of Director, Office of Alzheimer's Disease, as well as Associate Director for the Neuroscience and Neurophysiology of Aging Program at the NIH. Among other positions, he has been the founding director of Ronald and Nancy Reagan Research Institute on Alzheimer's disease. Professor Kachaturian has several publications in top journals relative about how to fight Alzheimer's disease. The title of his conference is Guidelines for Novel Conceptual Models of Dementia, Essential Features of an Ideal Theory, on Dementia. Professor Kachaturian, welcome to Spain.
I want to express my gratitude for the organizers for inviting me to speak at this meeting. I'm honored. I'm particularly honored by the fact that Her Majesty Queen Sofia is overseeing this conference. I'm particularly honored because she is one of the patrons for Alzheimer's disease, and for that I'm very grateful and acknowledge her contribution to the field. The main topic of my talk is really intended to change the thinking about Alzheimer's disease, because we're at a historic junction in Alzheimer's disease research. Although the disease was first described by Alois Alzheimer's in 1906, the modern era of biological studies began in 1961 in UK. It came at the Newcastle upon Tyne by a group started by Sir Martin Rode and the group that he put together. The very first meeting, scientific meeting, was in 1961 in that period. So that's the beginning of Alzheimer's research. In the United States, we began developing the program in the 70s. In that period, Alzheimer's disease was not well known. The number of scientists were only a handful, and the total amount of money spent for research was minuscule. Family members did not have any support groups. There was very little interest in the scientific community or public policy makers in Alzheimer's. Today, the picture has changed. We have close to 5,000 scientists around the world who are interested in Alzheimer's. There have been several networks that have been organized, such as the one in Spain, that the group you have. Similarly, in UK, in Germany, there is a network. In France, it's being organized. Many countries have begun to develop national plans. The disease has received the attention of policymakers. OECD issued an analytical report in 2013, which I prepared. The UK Prime Minister, David Cameron, convened the G8 summit that dealt with dementia. WHO has convened a meeting on. So the policymakers have begun to recognize the importance of the problem, because everyone knows that this is the mega problem for the 21st century, largely due to the revolution in the aging population. That is the revolution in longevity. People are living longer and longer. Many of the baby boomers are destined to live beyond age 100. Imagine if someone gets the disease at age 60, 30, 40 years ago, by age 70, most people died, so they had disability for 10 years. For the future, we're looking at disability for over 40 years. And that's going to cost tremendous amount to healthcare systems. That's why the burden is economic, social, scientific, and we have no choice but to solve the problem. Either that or be willing to ration healthcare or not allow people to live beyond a certain point because we can't afford it. Of course, there is an ethical problem with that solution. So the only solution is to invest into research and development to accelerate the technologies for early detection of the disease and technologies for prevention. So the major reason for my presentation is to find ways in which the scientific community could pick up the challenge and accelerate research and development to find ways in which we could slow or prevent this disease. One of the important issues is for this, in order to happen, we need to have a new thinking about the disease. And the need for new thinking is underscored by several factors. First, emerging knowledge regarding the biology of the disease shows that we're dealing with a complex and heterogeneous disease. So that's one reason why we need to think about the disease differently. Second reason is the failure of many of the clinical trials. During the last 20 years, many of the trials have not worked because they were based on current conceptual models of the disease, which have their limitations. Third reason is emerging knowledge regarding the biology of the disease requires a new paradigm for understanding the multiple interactions on a polygenic origin. The disease is not due to a single factor, but it's due to multiple factors. And we need to understand how these various etiologic factors interact along with many of the potential risk factors. During the last 10 or 15 years, we have identified several potential risk factors, including preventative, protective risk factors, such as education and so forth. So we need to understand how these interact in advancing the disease. Current treatments have not worked. They're not adequate. Right now, they work for about 2 years at the most. After that, the disease progresses and the patient is back to where they were. Therefore, the current treatments are not adequate for solving the large societal problem for the reasons that I mentioned, that the disease is going to last 30, 40 years of a patient. So we need to find ways in which we could ameliorate those conditions over that long course or prevent it from happening in the first place. Existing paradigms for drug discovery and development need to be rethought through. We need to find new ways of doing clinical trials in order to develop new therapeutic targets. So those are some of the reasons why rationale for the initiative of creating a new way of thinking, new conceptual models for the disease. Current models have failed to provide effective therapeutic targets for complex disease like Alzheimer's. Alzheimer's is only one example of complex chronic brain disorders. Whatever I say applies to other chronic late-life disorders. Alzheimer's is the prototype for it, but the same applies for Parkinson's, Huntington's, and so on. We need to account for the causal relationships between not only the biological phenotypes, but also the clinical phenotypes. So far, most of our treatments have focused on addressing the biological phenotypes, but have not failed to account for the clinical phenotype. So we need to understand what is the relationship between those two. Presently, no single theory encompasses all of the known, well-documented facts about aging and Alzheimer's disease. During the last 40 years, we've learned quite a bit about the brain aging and Alzheimer's disease. However, a lot of our knowledge is fragmented. They're not connected, so we need to find ways in which to connect. Part of the reason for launching this initiative of new conceptual models is to stimulate new thinking about underlying neurobiology of complex brain disorders, such as Alzheimer's disease, dementia, neurodegenerative disorders. A second reason is to sort out the complexities of the condition, to redefine the neuropathologic entity along with complex correlates of dementia. So the specific aim of this initiative is the way it's going to work is we have organized a work group. A work group consists of some of the world opinion leaders, including Professor Rossor, who is here, who have contributed to creating a format for the way in which we're going to solicit new conceptual models. The Alzheimer's and Dementia Journal of the Alzheimer's Association is going to devote a special section of the journal called Special Topics, devoted for considering perspective papers on new conceptual models. So I'll be describing for you the process by which that's going to take place. And we're going to invite the world scientific community to participate in rethinking through that process. So the specific aim of the special topic section is A, to create an inventory of ideas, facts, means, processes for comparing various theories, provide a knowledge management tool. We hope that eventually we'll be able to have all of the ideas, all of the pathways on a web-based program so that scientific community could start comparing different ideas from the amyloid hypothesis, tau hypothesis, inflammation, calcium hypothesis. What is the relationship? How the signaling pathways that account for each one of those are connected. So that would be a web-based tool to allow scientific community to compare and contrast. We hope to produce a common framework for considering different conceptual models, different ways of thinking about the disease. And eventually, we want to publish a comprehensive inventory of these different ideas. Shortly, I'll show you some examples of this. So the ideal feature for a unifying theory on dementia, mind you, right now, we do not have a unifying theory, so this is for the future. The ideal theory must account for both biological and clinical phenotypes. That is, you have to account for not only for the plaques and tangles, synapse loss, but also you need to account for how the cognitive changes occurs, depression, agitation, sleep loss. Those are the clinical features. What is the relationship between those two? Needs to be ironed out. Provide an explanation for a specificity of the neuronal system that's affected. The disease does not affect every part of the brain. It only affects certain structures, certain neurons. We need to understand why some parts of the brain are susceptible, why certain neurons are susceptible and others are not. And that specificity needs to be accounted for. We need to account for all the major risk factors in the disease, whether it be genetic, head trauma, education. There's a large number that have been identified. Another factor the ideal theory has to account for is the heterogeneity of the biological and behavioral signs of the disease. The disease is not a single disease, but appears to be a syndrome of multiple types. There's enormous variety. We need to account for that variation. Another factor is that the disease appears to start many, many years before the clinical feature appears. So there's a prolonged period when the disease starts. And we know very little about the very early asymptomatic stages of the disease. The theory needs to account for the relationship when it starts and how it progresses through into later life. And finally, the theory needs to propose testable hypotheses so that we can validate the ideas of whatever is proposed. Okay, here is an example of the heterogeneity of the disease. This is ability of the person to function. This is age, some arbitrary cutoff point for behavior. In some people in the population age quite well and they decline fairly late. Others cross this threshold point at various times. So we need to understand what causes that heterogeneity? Why? We know some of it. We know that certain genetic variables account for this. It might be there are some lifestyle variability that accounts for. We need to account for why this heterogeneity occurs, because if we understand that, that might give us therapeutic targets for treatment. Because if I understand what is the genetic and lifestyle variables that allow this individual to develop the disease at age 100 and they had dementia for 1 day or 2 days versus a person that developed it at age 60 and disabled for 30 or 40 years, that knowledge will give me a way to develop a therapeutic strategy in being able to do so. For that reason, it's important to sort out why this heterogeneity occurs. Most of the studies we've done so far during the last 40, 50 years has focused on the period after the symptoms appear, after the person develops the cognitive changes and so on. Now, the new era, we need to be operating in this area where before the symptoms appear. And that's the rationale for the OECD proposal to create the big data. That's the rationale for having your network, the network that's in UK, the network that's in Germany. Now people are beginning to follow healthy older individuals longitudinally so that we can find out what are some of these early changes that would allow us to predict what's happening. And that's a very, very important resource that needs to be developed. Now, during the last several years, we have developed several biomarkers. This is from Cliff Jack from ADNI. I'll go through very quickly. There are a large number of biomarkers. Some of them appear at very early stages in the cognitive normal stage, during the MCI stage, dementia. Most of these are developed on the basis of association with large populations. What we need to do now is translate that knowledge into being able to identify what is the causal relationship between these biological markers and the origin of the disease. In other words, we need to reverse engineer from the disease in what way some of these biological markers can be used as predictive models to allow us to predict accurately the likelihood of a person developing the disease in the asymptomatic stages. Remember, the payoff is going to be in these very early stages before the symptoms appear, where the person has not yet developed. Can we develop the new computational methods by having large numbers of samples, having a lot of information on genetic and other information that we have collected, into developing algorithm technologies for early and accurate prediction of who is going to get the disease. The big payoff for the field is going to be if we have the technology for accurately detecting the disease in a person at age 20 or 30 by having information on their profile and be able to track that individual through their lifetime. This is what I'm talking about. This is the pattern of the age. This is the various stages of neurodegenerative disorders. Very early stage, there are risk factors. Some of these are genetic, others are lifestyle. As time goes on, there's a biological marker, some indication, but there is no symptom. After time goes on, disability sets in, eventually the person dies. This slide illustrates everything. It illustrates the heterogeneity. Some people go through this course very early, others go later, others go much later. As I said, the most of knowledge we have now has been derived from this period. We're now trying to move into this area and be able to identify these very early risk factors in order to be able to predict which trajectory the person is going to take. Is the person going to be a healthy individual, live to age 120 without any decrement, or they're going to take a very rapid course or a moderate course? More importantly, what can we do to change the course? That's going to be the payoff. That's going to be the challenge. So that's the scientific challenge. In order to do that, we need to be thinking about the disease differently than we have done in the past. And that's the rationale for the presentation today. The ultimate aim of the disease is this. This is a metaphor for what's called synaptic reserve. This is a healthy individual. The person that's healthy has a neuron that has thousands and thousands of connections, but for some reason, variety of reasons, gradually as the disease progresses, they lose these contact points, and gradually it's diminished to that. That's, as I said, it's a metaphor from developing neuron. I'm using it as reversing backwards into Alzheimer's. So Alzheimer's is gradually moving from a neuron that's very healthy with a lot of connections into a point where it's not. And this really perhaps is the explanation for the clinical features we describe. Now, there are many reasons why a cell will lose neurons and synapses. It may lose it because of toxins. It may lose because of trauma. It may lose because of inflammation. It may lose because of toxins like amyloid, or it may lose it because tau protein prevents the exoplasmic flow. So there are a large number of ideas in the field of why this comes about. The problem is that none of these have really led to effective therapies, simply because the problem is a lot more complex than we've thought through. Here are some ideas, some of the ideas that have been around. Aging has been associated with the change that I described. Cholinergic factor was important, cytotoxicity, amyloid cascade, tau, my favorite, calcium hypothesis, inflammation, oxidative stress, and on and on. And here is some more. Genome-wide studies, epigenetic factors. So we have large number of ideas what causes the phenomena that I described, that is the loss of neurons, the loss of contacts. So in order for us to change our thinking, we have to introduce some new ideas. So one idea is a concept of complexity. I'll describe what I mean by that. The second idea is emergent behavior. And the third idea is systems theory. So I'm going to explain each one of these and then put it together into the framework for rethinking about the conceptual model of Alzheimer's. So let's first look at complexity. Very simple illustration of complexity. What I'm going to do is create a brain with 20 neurons. 1 to 20. This brain is going to have 20 neurons. If I have one neuron, and this brain is, all it's going to be required is to count. It's going to count from 1 to 1 million. That's his job. That's a very simple job, very simple brain. Now, if I have only one neuron, I can only count up to 1. I cannot count any higher. In order to count higher, I have to add another neuron. So I can count with 2 neurons, I can count up to 2. I keep adding neurons, and my ability to count increases. With 20 neurons, I can count to over 1 million. That gives me 1 million different combinations. Okay? Now look at the difference. Between 19 and 20, you have almost 500,000 complexity. That's an emergent complexity in the numerical system. The same thing happens in the, okay, here is again the same illustration. Between 19 and 20 neurons, there is not much difference in capacity to count. That's a very tremendous capacity. Now let's go look at the brain. This is the brain. As we add neurons, the ability of the brain capacity increases and reaches an asymptote, and then as it declines this way. As we take away neurons, it declines. The message here is that in a very complex system, a very small change can have a very, very large effect. So in a fully developed brain, a very, very tiny change, we're going to have a huge effect. The resolution required is enormous. Look at the resolution. That's the resolution. Here, 100% that. Imagine in the human brain where you have 13 to the 13 billion neurons, where you lose few neurons, you have a tremendous effect, yet you're not going to be able to see it. With current methods, we cannot measure it. So we need to develop technologies for detecting minuscule changes in brain circuitry, brain activity at a level that has an incredible resolution, the likes of which we don't have right now. So that's the picture of the complexity. Another issue is the emergent behavior. Now, with the brain, we start with a very complex behavior, and then we simplify that because understanding the brain is very difficult. So we simplify it, get it into lower and lower. This is called reductionistic approach. We start with behavior, in here we have clinical feature, and we studied the neuronal activity at molecular level. We identified the gene, and so on. Now, this is very useful for understanding the mechanism of complex biological system. In developing therapy, we try to go in the other direction. We go from the rat to the human. We do our experiments in therapy development on the rat with the idea that what it works in the rat or the mice, it's going to work in the human. And lo and behold, it doesn't work, simply because the model is not adequate. The reason it's not adequate is that there is the phenomena called emergent behavior. The clinical features we're dealing with in complex brain disorders are emergent behavior. They're non-linear. You cannot translate from here to there in the reverse way, in a linear fashion. The organization has different levels of complexity, and we need to understand how do you move from one level to the other. Let me illustrate another way. I may know everything there is to know about the water molecule. I may know everything about the physics. I may know everything about the chemistry. But that knowledge does not allow me to predict tsunami. Tsunami is, after all, due to water molecules, right? The same way in genetics, I may be able to know everything there is to know about the genome, but that doesn't allow me to predict a treatment. We've known the gene for Huntington's for many years. It hasn't been translated into treatment yet. I may know everything there is to know about the primary, secondary molecule of a protein, but it doesn't allow me to predict its folding characteristics or how it's going to interact with another protein, because that's an emergent characteristic of that system. So the concept of emergent characteristics is an important one, and we need to deal with it in developing the models that we have. And computational models, perhaps, is going to give us the ways to address that. Here is another way of saying what I was saying is we go from charged molecules to the complex behavior. This moving the therapy development from here to there has not worked because we don't understand the level of which the abstractions are organized at this level, how they translate into the next and next higher levels. We need to understand that transition points, and that's an important area of science that's not well developed. Okay, the third issue has to do with use of the systems biology, systems model, and I'm going to use that as a prototype example for rethinking of about the etiology of Alzheimer's disease. This concept is derived from systems theory, which requires for us to identify all the key etiologic components, and we have already done that. We have a large list of ideas that's involved. We need to understand sequence and interaction of these crucial variables. That we don't have it. We need to change the philosophy of how we do research, and that's very difficult. It's almost like having a religious conversion, which is in science, scientists behave like religious believers. When they believe in a concept, it's very hard for them to change their ideas. So the mindset of how research is conducted in pharmaceutical company needs to change, and that's going to require a heavy lift. Then we need to develop multiple strategies for developing therapies. A single approach may not be a solution, so that in order for the system to be corrected, you need to interact in a complex way. A metaphor for explaining this again is to look at the problem like a car. A Ferrari is a very complex system. Solution for optimizing its performance for a Ferrari may not be sufficient to just change the engine or improve the engine. You can get optimal performance from the engine, but if that engine is not coupled properly with the transmission or doesn't have the right kind of steering or the body shape, airflow is not correct, you're not optimizing the performance of that complex system. So the solution will require solving all of those problems at the same time. This idea is used in engineering, in solving problems like designing airplane wings, where you're looking at multiple factors, and the objective is to optimize the performance of that system. So what we're proposing here is in Alzheimer's, we have a systems failure. Neurons or set of neurons are failing, and they're failing for multiple reasons. And our job is to find out what are the key components that go into the operation, optimizing the operation of that system and improving that. And that requires different way of thinking, different way of approaching the problem. So dementia needs to be think about as a systems failure. And it's another important concept that it's not linear and unitary etiologic factor. That is, it's not caused by just amyloid. It's not just caused by cholinergic. That it's a combination of factors. There is a relationship between the cholinergic system and the metabolic dysfunction through the Krebs cycle. That's never been really studied. We know that if you deprive the neuron of glucose, you have a drastic effect on the nervous system. But cholinergic system gets selectively vulnerable because of that unique relationship. So in our modeling, we need to take that kind of knowledge and use the computer to see what would be the optimal relationship. So in developing cholinergic therapy, perhaps you have to combine it with metabolic stimulation. Just an example. Okay, now let's look at, I'm going to give you an example of looking at the problem from a systems point of view and using all the different ideas, these ideas that have been around. I'm going to use them, simplify, to show how we can design a systems approach to formulate our thinking about Alzheimer's. There are three major categories of changes that are required for maintaining the neurons healthy, to keep healthy. One is the energy requirements. We've known for a long time that if you deprive the neuron of glucose and oxygen, the cell dies. Well, you can do that through a variety of ways. You can do it through the mitochondrial defect or vascular changes or perfusion problems. We also know that the neuron's viability relies on very large array of signaling system, neurotransmitters, second messengers, gene regulators, endocrine factors. And then the third factor, unique to the neuron, is its ability to degrade and synthesize new proteins. The cell membranes, cell-embedded proteins are constantly being degraded and recycled and new ones made. That's part of the reason for the amyloid hypothesis, that it's a cell membrane that is a cell-embedded protein that gets degraded and it's recycled and there's problem. These three systems have to work in synchrony in order for the neuron to be functioning properly. So this idea of this going in this direction is due to one of those three categories of changes. Now let's make the problem a bit more complex. Let's add some other variables. These are the three systems. Now we ask the question, in what way aging affects those three systems, and in what way aging is different from a disease entity, whatever that disease might be. We want to know what is the effect of genetics. There's a large number of genetics on the various. We have only begun to scratch the surface. We have studied the genetics of how various protein, the amyloid, tau, and so on. So some of the genetics is known. Very little is known about the genetics of the signaling system, but that's another area. Genetics of various vascular or metabolic-related issues. There are immune changes that affect endocrine. This table sort of incorporates all of the ideas that have been around. The problem is that we don't know what the relationships are, and that's what we're looking for. We're trying to create a map, looking at the sequence of what comes upstream, what are some of the epidemiologically derived risk factors that come upstream, and what are some of the changes that are going on, what's due to aging, what's due to disease, and what are the downstream events, what are the consequences, so that you have a continuous functional flow. And the idea for the exercise that we want to launch over the next 2 or 3 years with the scientific community is identify all the different ideas, different theories, and look at the relationships, develop and put it on the computer so that we can do computer modeling and see if these things work and how it can be manipulated and so on. So that would be the general objective of what we're trying to do. And again, this is an invitation for the scientific community to participate. I sent the editorial that will be appearing in the next issue of the journal. Perhaps those of you who are interested can send me an email. I'll send you a copy of it or invite you to participate. So as part of the exercise, in order to make the problem easy, we have identified 10 key questions that each theory, each proposal, each conceptual idea model has to address. First one is risk factors. The important thing is, does the theory, whether it's the amyloid hypothesis or tau or the calcium or inflammation, does it address aging in what way does it address? Second question is, does it address risk factors, whether it be education or lifestyle, genetics. Second area is the progression and sequencing. How does the proposed proposal address the progression of the disease, going from the asymptomatic through MCI to mild and get into? What are the sequences? Which comes first? Does the metabolic changes, mitochondrial changes occur first, and then the neurotransmitter changes, then the protein degradation? What is the sequence of changes that occur? It has to account for mixed pathologies. We see that there is very pure Alzheimer's right now when you look at the neuropathology. Always there's various components, so we need to account for the mixed pathologies. And it needs to account for the multiple clinical phenotypes, such as FTD, PCA, frontotemporal dementia. All these have to be accounted for by the new theory. How does the theory account for the biomarkers? I showed you the ones that have been identified through ADNI, and some others are being identified. Does the theory account for this, and does it account for the sequencing? Finally, does the theory allow us to develop a testable hypothesis so that a therapy could be developed? With that, I'll close. Again, I want to thank you for inviting me. I want to express my gratitude to Her Majesty the Queen for attending and patiently listening to this babble that I'm giving. Perhaps nothing made any sense, but I hope something made sense. Again, thank you.
Kachaturian, for your excellent lecture. And I think that we are, with a little delay, we are out of time already. Perhaps we can stop here. We will not have a question for you, if you don't mind. Okay, and we will continue with the program. And the program is that afterwards we will move to see the posters and to move to outside, prepare for the lunch, if you don't mind.
I kept the right ones out and let the wrong ones in. Had an angel of mercy to see me through all my sins. There were times in my life when I was going insane, trying to walk through the pain. And when I lost my
Okay, we are going to start. Please take your seats. Okay, we will continue. We will continue with the next speaker, Professor Martin Rossor. It is a pleasure to introduce the talk of Professor Martin Rossor from the Institute of Neurology in London, UK. Professor Rossor is currently the president of the Association of the British Neurologists. Professor Rossor is also the director of the British National Institute for Health Research in the area of dementia. The work of Professor Rossor is related to the use of novel neuroimaging methods for the diagnosis and monitoring of neurodegenerative dementias. He has been involved in several clinical trials. Professor Rossor has an excellent CV in the field of dementia, where he has a relevant publication in top journals. These works are very well known in the area of Alzheimer's disease, where Professor Rossor is a well-recognized scientist. I like to meet Professor Rossor in these circumstances because previously I remember once that we met in Stockholm. At that time, a volcano in Iceland was active, and we were unable to return home by plane. And I remember that finally we did by taxi. That was not very pleasant. The title of Professor Rossor's talk is Responding to the Dementia Challenge. Is there a window of opportunity with early intervention? Professor Rossor, welcome to Spain.
Jesus, thank you very much. I don't really need to tell this audience of the importance of dementia. And could you put the right hand? There we are, thank you. Of the importance, not only the human tragedy of dementia, but also the enormous economic cost, calculated as being equivalent to the gross national product of Turkey. So enormous. And that's not just the cost itself, it's the opportunity cost, it's the research that we can't do into other problems that we can't do to solve things like global warming, and the cost to our grandchildren in that economic burden. The political drive has been very valuable. The UK used its presidency of the G8 to create the Dementia Challenge and have a G8 Global Dementia Summit, and a number of countries since then have carried forward that initiative, and it is essential that we continue to do that. And the WHO sort of finished off earlier this year that first tranche of political activity with a ministerial conference in March. There was a declaration at that G8 summit, much of it, I think, fairly obvious of what one would aim at. Of course, it's important that we can't trade off care today for research tomorrow, but the very first bullet point is perhaps the key one, and that's the one that Zaven Kachaturian spoke to. Can we be identifying a cure or a disease-modifying therapy? Now, it's interesting the term dementia was used there. Politicians tend to conflate dementia with Alzheimer's disease, but of course most of the focus at the moment is inevitably on Alzheimer's disease. and we haven't done very well. So this was a review of what was in the pipeline in 2010 in this nice review in the Lancet Neurology. All looks quite encouraging with that pipeline of drugs, but most of those that were going into phase 3 have since been killed off. Some of them are perhaps coming back as we change our view of when we do need to treat. But when you look at the drugs that are available for Alzheimer's disease, and I won't only be talking about Alzheimer's disease, it's over 10 years since anything has come onto the market. Now, Zaven's already mentioned that much of this may relate to the fact that we are dealing with patients who fulfill the criteria for Alzheimer's disease, and although there's been a shift more recently to how we might define that entity, until relatively recently, it's required somebody also to fulfill the criteria for dementia, i.e. you are waiting until something is very severe. It's like waiting until a neoplasm has metastasized before you decide that you can make a diagnosis and start treatment. Doesn't quite feel right. And so this is, just to illustrate that, this is from ADNI data just showing that if you take individuals with established disease, then in terms of brain shrinkage and ventricular enlargement, you are dealing with a very far advanced and rapidly advancing disease state. So one can't really rely on doing your trials at that stage. And I think we do have a problem with this term dementia. It's politically very useful, but to wait until something is sufficiently severe is, I think, problematic. It's partly to do with how we interrelate with patients. It's partly to do with the conceptual difficulties we had with these diseases until relatively recently. Of course, dementia is only the tip of the iceberg, and underneath one assumes, early progressive disease and pre-manifest disease, be it Alzheimer's disease or Huntington's or vascular. And this term mild cognitive impairment, which is of course not a stable construct, it just describes something that's slightly less severe than dementia. And of course there are numerous causes for people having some impairment of their cognition. But that stage of pre-manifest Alzheimer's disease or early Alzheimer's disease has been around for a very long time, and particularly John Morris and colleagues and Price have provided suggestive evidence from cross-sectional post-mortem studies of individuals suggesting that quite a long time in elderly people before you may get the manifestations of significant cognitive impairment, one has variable degrees of the pathologies that we associate with plaques and tangles. So can we learn more about these pre-manifest stages of the disease? Can we understand this to move trials, as Zaven suggested, into those earlier stages? The problem is that pre-manifest Alzheimer's disease, and I'm just illustrating this with Alzheimer's at the moment, pre-manifest Alzheimer cohorts are very difficult to come by. You can, of course, go for population-based, and scientifically these are very good because these are naturalistic populations, but you do have to examine a very large number of people in an aging cohort in order to pick up those relative number that will go on to develop a disease, and so you're limited by how detailed an assessment you can do. Mild symptoms, you're probably already well down the stage of the disease. You can enrich by age, you can enrich by genotype, but probably the most detailed information has been in those with autosomal dominant disease. For Alzheimer's disease, those who carry mutations either in the presenilin 1 or 2 genes or in the amyloid precursor protein, the APP gene. And the dominantly inherited Alzheimer network run from WashU has brought together a very large number of centers, predominantly in the US, who see individuals with those familial Alzheimer's disease genes, often coming on in their 30s, sometimes 40s and 50s, and have been able to infer from the cross-sectional data that there is indeed a long pre-manifest stage. And this is a summary taken from Randy Bateman's New England Journal of Medicine paper, suggesting this is rather very much a suggestion that the A-beta 142 may be high early on, but actually the drop in the CSF changes are really quite early. CSF tau and A-beta deposition, the latter being measured on PIB scanning, later hippocampal volumes and glucose metabolism, and then much later the clinical features. The A-beta deposition, I just show this slide because it is an example taken from Diane. Here's an individual who carries a mutation. These are the non-carrier individuals that, of course, provide extremely good controls because they've had early environmental exposures that are common, being brought up together, and they will share much of the genome apart from the specific mutations. Just one point of note, though, is the prominent striatal deposition using PIB scanning. I do just draw attention to that because we are making the assumption that you can generalize from familial Alzheimer's disease to the more common later onset Alzheimer's disease. One cannot assume that. Much may be generalizable, but these may be quite different diseases. And I think that the pattern in PIB suggests that something rather different may be going on. But from these data, the WashU group has suggested a sequence of events in the development of Alzheimer's disease in terms of the biomarkers, that you pick up changes in A-beta in the CSF first, and then you get the deposition of amyloid fibrils with PIB binding, increase in the tau and hippocampal volumes going down before you get the symptoms. This is an inference. This is, of course, an inference that's similar to the Braque and Braque inference, which are only cross-sectional data, and there are problems with assuming that that's going to be the sequence of events. And there is also a problem in that you estimate when you think symptoms are going to come on by comparison with the parents, comparison with other family members. And of course, diagnostic approaches, people's perception of disease changes. So it's likely that if you're looking at cross-sectional data, you will assume or infer that there are changes earlier than they really are. And in order to get some sense of what happens in the real world, rather than inferring from cross-sectional data, you do need to follow individuals for a long period of time. Now, that's very expensive, very difficult. So I just illustrate a couple of examples of individuals because I think it helps. So this is somebody with a presenilin-1 mutation, and these are volumetric T1 scans taken over a period of 6 years. And at time 0, the individual was entirely well. If you, however, register the scans to the baseline, you can see the ventricles are actually rather larger here 6 years later. You can see that there has been shrinkage. This is the point where they began to develop symptoms. And this just shows this in a little bit more detail. So this is the brain volume. This is symptom onset. This is actually when the person fulfilled the original NIHR criteria for Alzheimer's disease. Sadly, this person who developed the disease at 36 subsequently came to post-mortem and had typical Alzheimer histopathology. These are fluid registrations against baseline, where red indicates expansion, so the ventricles are expanding, and the green and blue indicates shrinkage and atrophy, which, as you can see, are here in the medial temporal lobe and then into the association cortices. And just looking at that at a group level, now just going to show you are a relatively small number of individuals like that, where one's actually looking at where the rate of change differs, because it's the rate of change that one's interested in. And if one looks at pre-symptomatic mutation carriers, you can see that in fact there are changes here in the medial temporal lobes and in the association cortex in the cingulate area. This is rate of change. So this is where the rate of change differs from the rate of change that might occur with aging in the mutation-negative cases. More extensive when you look at the mildly affected, and when you get to the stage where you actually have established disease. In fact, interestingly, it's more extensive, but you lose that significant signal in the medial temporal lobes. Remember, this is atrophy, so it may just be there's a lot of variance, so you don't see a statistical significant difference, but it may actually be that once the disease has progressed a certain stage, in a sense it's burnt out in the hippocampus, and you haven't got a lot more to lose in that area, so you won't see a signal when you're looking at the rate of change. This does look, though, rather like a Braque and Braque inferred map of the progression of the histopathology, and provides some assurance that at least in familial Alzheimer's disease, this is a prolonged pre-manifest stage of the disease, which should be the target for treatment. And there are now a number of trials have been started in this particular group. There's the trial in the Colombian presenilin 1 kindred. There is the dominant international Alzheimer network trial, and then there's the A4, the asymptomatically elderly patients who have PET amyloid positivity, all with different immunotherapy approaches. The DIAN trial has an adaptive design, so based on biomarker change, and if there's no evidence of target engagement, then the drug is dropped and the next one becomes available. If there's evidence, then they'll move on to be looking for cognitive and behavioral changes. I do think this is going to be critically important. It may be that you won't be able to generalize from something that is able to treat familial Alzheimer's disease into the more common later onset disease. But at least once you've got something that may be effective, then you can start designing better trials, and you can begin to understand how to approach the disease, because you've got a gold standard, you've got something that you know will actually work. As Zaven said, it's not all about Alzheimer's disease, lots of other causes of degenerative dementia, and much has been learned recently about the frontotemporal lobar degenerations, that intriguing group of diseases divided up to the behavioral variants, extremely difficult to manage in the clinic, and those with language and speech problems, and divided between those with a genuine language, a breakdown of their semantics, versus those with speech production difficulties. And it is very complex, and don't need to go into this in any detail, but other than that, here are the various phenotypes that one sees in the clinic, here are the underlying molecular pathologies, but importantly, these three genes, C9orf72 expansions, granulin, the progranulin gene, and microtubule-associated protein tau, are the main contributors to familial frontotemporal dementia. And a autosomal dominant family history is probably found in about 15 to 20% of individuals. So this provides another opportunity to explore whether there is a pre-manifest stage and how one might best approach treatment. And to look at that, there's been a genetic FTD initiative based in Europe, linked in with Canada, using EU money and local national money. It's run out of University College London, and the centres that are in capitals there are those that have joined the GenFi initiative just in the last year or two, and very good to have Barcelona and San Sebastian have now joined to contribute families with NERN, tau, granulin, or C9orf mutations. And one can make the same theoretical progression of biomarkers that one sees with Alzheimer's disease, the famous Jack curves. And yes, there is evidence that this applies to frontotemporal dementia as well. These, I would again emphasize, are only cross-sectional data, and therefore it is likely that they overestimate how early the disease begins, but this is just looking at cortical volumes against the anticipated age at onset here, and the insula appears to be common and early across these different diseases. There's a problem at the moment. These are only the first cut of data. These was published in Lancet Neurology earlier this year, in that because of the relatively small numbers, all of this group of diseases were in fact grouped together for these analyses, but there are now emerging some differences, in particular around tau and its early involvement of the medial temporal lobes. And I'll just show an example. Again, these individual examples are hard to come by, but they're very informative. So this is an individual with a tau mutation, and again, this is the fluid registration showing the striking symmetric anterior temporal lobe and hippocampal atrophy. And these are the hippocampal volumes of this individual against their sibling who did not carry the mutation. And you can see here that, in fact, changes are being picked up about 8 to 10 years before symptom onset. The advantage here, of course, is you're following people through until they actually develop the symptoms, so you know exactly what the natural history and the progression of the different biomarkers and the appearance of the behavioral changes are. And just to show you, we've been able to just begin to undertake PET imaging with the AV1451 tau ligand, and this is again a case of a tau mutation where the tau signal very much mimics those areas of atrophy that you saw in the anterior temporal lobe. Progranulin is very different, and I always like showing this video of registered volume MRI images because of the striking asymmetry that occurs with progranulin mutation-positive FTD. I don't know the reason for it, whether it's something to do with some asymmetric insult because of the role of the granulin molecule. I'll just show you, this is somebody who was imaged over a number of years and across the time that they actually developed symptoms. And if you see on the left there, or the right facing you, just how strikingly asymmetric and rapid the atrophy is in these progranulin cases. So yes, it does suggest that there is this very important pre-symptomatic stage in these dementing diseases, perhaps as long as 10 years, where we should be directing our attention in terms of the research. It's quite different from arguments about screening people as a public health measure. This is about where do you target trials of interventions. But I would like to finish by suggesting that we may need to broaden our view around what's beneath the dementia iceberg. Zaven said there's many disorders other than Alzheimer's disease. And importantly, because we focus on dementia and its severity, there is an enormous amount of cognitive impairment that's beneath the waterline. And many, many things that we know will alter cognition. Medications, for example, general systemic ill health, stress, injury, pollution, poor education, and poverty. And that's only just a few exemplars. And all of these, of course, will impact upon the cognitive capital of a society, of a group, of a nation, or of an individual. And last month in the Lancet, Martin Knapp from the London School of Economics and I suggested in the Lancet that is it possible to adopt an idea from the other big global challenge we face, global warming, and actually model a cognitive footprint. And a cognitive footprint would either be at an individual level in terms of activities that people undertake, or would be at a group, a family, a group, societal, national level in terms of policies. And a cognitive footprint could either be positive in terms of benefit or negative in terms of a negative impact on cognition and cognitive capital. So that one can then return to some of these, and in thinking of ill health, it means that one can then begin to model sometimes those disorders that we forget about that are so important in developing countries like childhood malaria or neurocysticercosis. Modest but measurable changes on cognition. And of course, negative cognitive footprint or negative impacts early in life will have a bigger cognitive footprint than those that may be emerging later in life. One can be aware of the problem of poor education and poverty. The introduction in the UK of raising the school leaving age in 1947 has impacted upon, it is believed, an effect on robust resistance to cognitive impairment in later life. Medications. We are very used to patients not taking medication because it makes them feel sleepy or not with it. And we tend to ignore these negative cognitive impacts of medication. Should we, in fact, be measuring a cognitive footprint of medication, a very simple real-world cognitive measure as part of the adverse event recording that then allows one to assess a cognitive footprint. But that's a suggestion. I personally think we do need to broaden the debate for looking ahead and see if it is possible to model cognitive footprints. And I think this is also at an individual level rather than just at a larger society level. So at an individual level, I always like to end with one of the Rochefoucauld maxims. It's, I'd like to die as young as possible, but as late as possible. I'll leave it there.
Thank you very much. I think that we have time, if you like, for some questions. I will ask if anybody has some questions. If in other case, I will start, if you don't mind. You saw us about the symmetry or asymmetry in cases like, for example, frontotemporal dementia and tau pathology is symmetric. But in the case of granulin, is asymmetric. Do you have any idea about if it's also any asymmetry for Alzheimer's disease, for any marker?
Yeah, I mean, yes, I think there is asymmetry, and I think one can see this in imaging. I think one can see it also clinically, but it's probably easiest to see in those slightly atypical or what we would think of now as atypical Alzheimer cases. So for neurologists, I think the posterior cortical atrophies can be very asymmetric. So you can have one visual field in which there's visual disorientation that does not occur in the other field. And that's the sort of posterior cortical atrophy presentation of Alzheimer's disease. So it does exist. Sometimes I think at a clinical phenotype level, it's what we can measure sometimes gives us an impression of asymmetry that may be false. And that may be the case, for example, logopenic aphasia as a presentation of Alzheimer's disease. But some of these diseases, I don't think I've ever seen anything as asymmetric as progranulin. It's just so striking. I don't know why.
Okay, another question. Okay, perhaps we are a little late, and then we will move to the next. Okay, this speaker is Professor David Rubinsztein from the University of Cambridge, UK. Professor Rubinsztein studied medicine and did his PhD at the University of Cape Town, South Africa. Afterwards, he moved to Cambridge in 1993 as a senior registrar in genetic pathology. Now he's working in cell and molecular biology. The work of Professor Rubinsztein deals with two words, autophagy and neurodegeneration, using Huntington disease as a paradigm. Huntington disease is a proteinopathy with toxic protein aggregates and with another amount of aggregates. Professor Rubinsztein Laboratory was the first one to suggest the strategy of autophagy upregulation as a possible therapeutical approach in various neurodegenerative diseases like Huntington disease, and has identified drugs and novel pathways that may be used for this objective. Professor Rubinsztein has an excellent CV with papers with many citations and published in high-impact journals. Professor Rubinsztein is a fellow of the British Academy of Medical Science since 2004, and he has been awarded with several international prizes. He's now at the top of his field, and he will talk us today about autophagy and other therapeutical strategies for neurodegenerative diseases. Professor Rubinsztein, welcome to Spain.
Thank you for the very nice introduction and for the invitation to be here. It's good to see old friends and to make new friends. I didn't plan my talk in relation to Martin's, but this is my first slide. And I think, normally I start my talks with the patient, but I think it's better to start the talk with the success story. So this is one of my heroes. When I'm not working, I play the cello for fun. And Pablo Casals was a great Spanish cellist. In fact, he's one of the greatest cellists and musicians of all time. And he's my hero also because he was a great humanist. But he's on this slide because he was like Gentleman Martin's last slide, in that he's a real example of successful aging. He lived into his 90s. He was active into his 90s, as you can see on the slide. And in fact, he married for the second time in his early 80s to a woman who was 60 years younger than he was. That type of longevity, healthy aging, is what I think we all aspire to. So how are we going to get there? And today I hope I'll be able to give you some of the hints of the types of strategies my lab's been using to see if we can stave off some of the diseases that prevent healthy aging. I'm going to divide my talk into 4 sections, but really only into 2 parts. In the first, I'm going to describe our older work relating autophagy to neurodegeneration, and then describe how this led us to search for new signaling pathways that regulate autophagy. Finally, I'm going to tell you about what I think is an important component, is that how we can link basic science related to the cell biology of autophagy to understanding disease pathogenesis. So how our basic science has given us insights into disease pathogenesis. And then in the final part of the talk, I'm going to tell you something completely different to autophagy, which might also end up being a druggable pathway that we can exploit to stave off neurodegenerative diseases. So for this audience, I don't really need to rehearse the fact that most adult-onset neurodegenerative diseases, and indeed childhood-onset diseases like lysosomal storage diseases, are associated with the formation of inclusions or aggregates within the cytoplasm of neurons. And there's extensive genetic and transgenic evidence that suggests that these types of aggregate-prone proteins, particularly in Mendelian forms of these diseases, exert their pathogenesis through toxic gain-of-function mechanisms. So the strategies that we've been interested in over the years are either to find ways of enhancing the ability of cells to get rid of these types of poisonous proteins, just lowering the load of the poison for the cells, or alternatively, to find ways of helping those proteins fold so that they're not so aggregate prone. As the first part of the talk is about autophagy, let me just give you a quick tour through what autophagy really is. The first morphological characteristic feature of autophagy is this cup-shaped double membrane structure called a phagophore that forms more or less randomly in the cytoplasm of most cells. And as its edges expand and then fuse, it engulfs a portion of cytoplasm containing whatever happens to be in that cytoplasm. These autophagosomes are then trafficked along microtubules to the microtubule organizing center of cells where the lysosomes are clustered. And this enables the fusion of autophagosomes and lysosomes and the subsequent degradation of the autophagic contents by the lysosomal hydrolases. About 12, 13 years ago, Brinda Ravikumar in my lab found that if one slowed the formation of autophagosomes or the fusions of the autophagosomes and lysosomes, one slowed the clearance of the oligomers or the aggregate precursors for mutant Huntington. And in doing so, it increased the accumulation of these proteins both in the soluble species and the aggregate species, and in conjunction, this enhanced cell death. And we showed this initially in cells, but subsequently in Drosophila and mouse models. But what was perhaps more interesting about our findings at the time was that if we enhanced the clearance of these proteins in cells by upregulating autophagy, we could ameliorate toxicity, either in cell models, neurons, Drosophila, or mouse models of Huntington's disease. And this was accompanied by decreased accumulation of the mutant proteins. The important point to make is that when we initiated these studies, we used rapamycins because they were the only drugs we were aware of at the time that were predicted to induce autophagy and were used chronically in human conditions. We've expanded the list of diseases where there's potential autophagy upregulation to a wide number of neurodegenerative conditions. As you all know, Huntington's disease is the most common of the 10 conditions caused by expanded polyglutamine tracts in the protein. And the second most common is a disease called spinocerebellar ataxia type 3, or Mercado-Joseph disease. And we showed in cell-based systems and in mouse models of this disease that the mutant protein is an autophagy substrate and that we could virtually normalize the phenotype in a mouse model of this disease even if we initiate the treatment after the initial appearance of signs. In addition to the 10 polyglutamine diseases, there are 9 diseases caused by abnormally long polyalanine tracts in the protein, and these proteins are also aggregate prone, and as a model system we showed in cells and in Drosophila models that such proteins were autophagy substrates and that there would be benefits to autophagy upregulation. But perhaps the most important diseases on the slide are represented by those involving tau, where wild-type tau is believed to be a mediator of pathogenesis in Alzheimer's disease, and point mutations give you frontotemporal dementias. And we showed that in cell-based systems that both wild-type and point mutants of tau are autophagy substrates, and that in Drosophila we could ameliorate the phenotypes by upregulating autophagy. And finally, alpha-synuclein is the protein that accumulates in Parkinson's disease, and we showed that its clearance could be enhanced by autophagy upregulation. This idea has now caught on in the field, and many have now tested this concept in their favorite models. And this slide just gives you a hint of what's been going on in the field, and you're not supposed to read the details, but just see that there's been a lot of activity and validation of the key concept in rodent models of many of these diseases. So our spinocerebellar ataxia type 3 data being replicated, there are data in Alzheimer's disease, tauopathies, Parkinson's disease with alpha-synuclein overexpression, ALS or FTD with TDP43 mutations, and even in a familial prion mutation in mice. There is a health warning on the slide, though. So although the concept might appear that autophagy might be able to solve all these diseases, I don't believe it's the case. I think autophagy upregulation might be a viable therapeutic strategy where you've got a condition caused by an aggregate-prone protein, but where autophagy flux is not impaired. And by that I mean that there's not any delayed traffic of the autophagosomes to the lysosomes, because if that were to occur, so if autophagosomes aren't properly removed by lysosomes, then upregulating autophagy is just going to lead to an accumulation of autophagosomes, and that might be unproductive or even toxic. So I think the message is, before thinking about treating a disease with autophagy upregulation, one really needs to understand the biology of the system in the relevant disease condition. I mentioned earlier that when we undertook our initial studies to demonstrate proof of concept for this idea, we used rapamycins, but it became apparent to us that we wanted to treat patients from the earliest possible age. So using Huntington's disease as a paradigm, we would want to treat patients who are gene positive, but perhaps from the age of 18, 19, with the idea of actually delaying the onset of disease and buying them more years of healthy life. So we need to find drugs that we can give people for decades that are going to be well tolerated and safe. And to that end, we've undertaken a number of chemical screens and identified a number of lead molecules. And I'm just going to tell you about one set of molecules that we identified from a screen that we undertook with a library that was highly enriched in compounds that had been used in man previously. So these are FDA-approved drugs, and we are basically looking for novel uses for these drugs as autophagy upregulators. And the drugs shown on the slide are all autophagy inducers. They enhance the clearance of aggregate-prone proteins like mutant huntingtin or forms of alpha-synuclein, and they mediate protection in cell, Drosophila, zebrafish, and mouse models of Huntington's disease. And this is a complicated slide because these drugs act on a pathway where there are connections between the key nodes, and I can talk you through that quickly. So some of the drugs act on these imidazoline receptors that decrease cyclic AMP, and this ends up inducing autophagy. Cyclic AMP blocks autophagy by activating this EPAC protein, which is a guanine nucleotide exchange factor for the small g protein called RAP2B, which in turn activates phospholipase C epsilon to produce too much IP3. IP3 blocks autophagy by a number of mechanisms, but one is by binding to receptors on the endoplasmic reticulum, releasing too much calcium, and the calcium we found activates calpains, which is known to occur, but calpains block autophagy by cleaving this GS protein to constitutively activate it and in turn form more cyclic AMP. So we've got drugs acting on multiple nodes of this pathway, on the imidazoline receptor, on the L-type calcium channels, on inositol triphosphate metabolism, and approaches working on calpain. And we validated all of these in mouse models of disease. One of these we've already taken through to a clinical safety trial, rulminidine, which acts on the imidazoline receptor, and this is just about complete and is being driven by Roger Barker in Cambridge, my colleague, looking at early-stage Huntington's disease patients. I just want to give you a flavor that autophagy is a protective strategy. It's not something that's confined to neurodegenerative disease. My good colleague, Vojo Deretic, showed that autophagy can enhance the clearance of mycobacteria, and he did his experiments in cell culture. So we wanted to see whether this was the case in vivo. And so what we did is we took all the drug hits from the previous slide, and we looked to see if they could enhance the cellular-based killing of mycobacteria. The drugs themselves have no effect on mycobacteria viability in isolation, but they enhance the ability of cells to kill the mycobacteria. So these experiments are done in mouse macrophage cell line, and then we took some of these drugs further into primary human macrophages and human alveolar macrophages. And the drug we want to concentrate on is the anticonvulsant carbamazepine, where we found that it had efficacy at concentrations that was similar to those that would be seen in patients taking the drug. And therefore we collaborated, and so this work is done in collaboration with Andres Floater and Diane Ordway, and Diane Ordway did the mycobacterial work in mice. And so what she did is she infected mice with multi-drug resistant mycobacteria, and the back shows the placebo-treated mice. The blue shows the mice treated with the conventional chemotherapy for tuberculosis, and it has no effect because these are drug resistant. But the carbamazepine has quite a dramatic effect, especially given in mind that there's a log scale on this axis. So we feel that we have broadened the repertoire potentially of autophagy upregulating drugs to this class of diseases. And I think going to one of the points Martin made earlier, when I was a medical student in South Africa, I saw patients with cerebral tuberculosis. So there's even a reach for these types of strategies into brain diseases. But rarely to veer outside the field. I'll tell you a quick vignette. One of these outstanding postdocs that's just left my lab is a man called Kevin Moreau, and I'll show you some of his work just now. And he had a brother who was doing his PhD in a lab working on Pacific oysters. So the two brothers were talking, and the upshot of the discussions led to a collaboration where we showed with his lab, Tristan and Renault's lab, that autophagy upregulation, indeed carbamazepine, can protect oysters against both viral and bacterial toxins. So the reach of autophagy can even go into food production if you're interested in shellfish. So that brings me to my first conclusion, that autophagy enhancement might have benefits by reducing the loads of these poisonous proteins, which are aggregate prone, and various pathogens. We've also shown, and I can describe the mechanisms, that it reduces susceptibility to a range of pro-apoptotic insults. As I've shown you, we and now others have shown that autophagy upregulating strategies are protective in a range of cell and animal models of Huntington's disease and related conditions, and these benefits can be mediated by drugs acting on the target of rapamycin or by mTOR-independent strategies like I showed you with the FDA-approved drugs. With that in mind, we've made a big effort in the lab to try to understand as much as we can about autophagy signaling with the aim of identifying new therapeutic targets. And I don't have time today, and you're probably not interested, to go through all the detailed cell biology. But suffice it to say that the biology of the target of rapamycin is rather complex, and we've done quite a lot of work around it. And many of the genes that we've identified that regulate autophagy are involved in cancer. And so there's an additional component to autophagy that is outside my lab's main interest, but that is in the context of carcinogenesis. But it serves as a segue into telling you about our discovery of a non-canonical autophagy signaling pathway that I believe reveals a new therapeutic target for neurodegenerative diseases. So canonical autophagy signaling is driven by a lipid called phosphatidylinositol 3-phosphate. I'm just going to say PI3P so I don't get tongue-tied. And this drives autophagy by recruiting a protein called WPI2, which in turn recruits proteins like ADG16, which dictate the site of autophagosome formation on specific membranes. PI3P is generated by a kinase called VPS34, and that is canonical autophagy. So VPS34 makes PI3P, which dictates the sites of autophagosome formation. But there have been papers in the literature suggesting that you can get VPS34 independent autophagy. So if you knock down or knock out VPS34, you can still have autophagy. And this has been observed particularly in cells that are glucose-starved. So one possibility is that there's a VPS34 independent source of PI3P, which there might well be. But what I'm going to tell you today is that VPS34 autophagy or PI3P dependent autophagy can be substituted by a different lipid. And that is the mirror image phosphatidylinositol called PI5P. And this is work that was driven predominantly by Mariela Vicinanza in the lab. So the first set of blots show experiments where we've taken cells and loaded them with PI5P or the carrier lipid. And so I don't know how well you can see the blots, but this band called LC32 correlates with the number of, or the volume of autophagosomes in the cell. And you can see that the volume goes up. This is a light and a dark exposure of the Western blot. It goes up very dramatically when you load with PI5P. If we want to measure the rate of autophagosome formation, we clamp their degradation by adding a lysosomal inhibitor like baflomycin A1. So any changes in the autophagosome number reflect changes in formation. And you can see when we treat cells with baflomycin A1, BAF, you get an increase in the number of autophagosomes when you loaded PI5P, suggesting that you're getting more autophagosome formation in that context. If we assess using cytometric methods the number of autophagosomes, the number of autolysosomes in the cell, you can see that they go up in a dose-dependent fashion when we load the cells with PI5P. So the autophagosomes are in yellow, the autolysosomes are in red. So that's loading cells, and that's an acute strategy. But we wanted to understand the genes that regulate autophagy through this pathway. So PI5P sits here in the pathway. It can be synthesized from phosphatidylenosytol by an enzyme called PIC5, and we used chemical inhibitors of this enzyme to show that it acts as one would expect in the pathway. It can also be synthesized from PI3,5P2 by removing the phosphate at the 3 position by myotubulins, and we've shown that myotubulins play a role in this pathway as well. But the enzymes I want to focus on are called PI5P4 kinases, and it comes in 3 forms. There are 3 isoforms of these enzymes which all behave in the same way for us. So these enzymes change PI5P into PI45P2, and so they're basically metabolizing the PI5P. So if we knock down these enzymes genetically, we accumulate PI5P in the cells, we increase the number of autophagosomes, as we can see by the LC3, and we increase autophagosome formation, as you can see in the presence of the lysosomal inhibitor, and similarly, one increases the number of autophagosomes and autolysosomes when one knocks down these enzymes. I should point out, so I don't have time to show you, if we overexpress any of these enzymes, we get the converse effect. So our idea was that PI5P is the mirror image phosphoinositide to PI3P, so we want to work out if they are functionally interchangeable. So what we did is we took cells and we looked at the effect of the VPS34 inhibitor Wart-Manon on these cells, and as expected, it decreases PI3P and decreases the number of autophagosomes, in fact, the rate of autophagosome formation. And this can be rescued when you take the Wart-Manon treated cells and you add back the PI5P. So this suggests a functional rescue. We confirm these effects by using genetic strategies to increase the PI5P levels, for instance, by using knockdowns of the PI5P4 kinase enzymes. We confirmed the autophagic data by electron microscopy, and we showed that the PI5P effects with Wart-Manon were also seen if we used an alternative strategy by knocking down the VPS34 kinase. So we did a lot of experiments, which I've just shown you one, that suggests that PI5P can rescue the effects of PI3P depletion. So we did the converse. We showed that PI3P could rescue the effects of PI5P depletion. So how are they working? So I told you earlier that canonical autophagy works by PI3P binding to this protein called WPI2, which then sequesters proteins that are important for dictating the site of autophagosome formation. So we used biophysical approaches to try to show that PI3P and PI5P could both bind and compete for binding for WPI2. So what we did is we took cells and we transfected them with GFP-tagged WPI2, and then we took beads that were either uncoated or coated with PI3P, PI5P, or PI, and looked to see if the beads could pull down the GFP-tagged WPI2 bead. And here I show the pull down with the PI5P and the PI3P. In order to show that this effect was specific, we did the experiment with PI5P and showed that the effects of the PI5P coated beads could be competed out by incubating the mix with liposomes containing PI5P. Similarly, the effects of the PI3P beads could be competed out by liposomes containing the PI3P. But then, importantly, we showed that the effects of the PI5P could be competed out by beads by liposomes with PI3P. So the PI3P and 5P compete. And similarly, the beads with PI3P can be competed out by beads containing PI5P. So biophysically, they can compete for binding to the same substrate. Finally, the domain for binding WPI2 to PI3P is being inferred, and we mutated the domain and showed that this reduces binding to the PI3P beads, and it reduces the bindings in a similar way to the PI5P beads. So, that gets me to a model. This is PI, this is PI3P, this is PI5P. They're mirror image phosphoinositide. And they both play similar roles regulating autophagy by being attached to key membranes, where they recruit proteins like WPI2, which in turn recruits ATG16, which dictates the sites of autophagosome formation. They're functionally interchangeable with normal autophagy and with starvation-induced autophagy, with full starvation-induced autophagy with amino acids. And as I've shown you, they are also functionally interchangeable in that the effects of PI3 depletion can be rescued by PI5P and vice versa. Interestingly, we showed that glucose starvation-induced autophagy is independent of PI3P, but dependent on PI5P. But this brings me to what I believe is the therapeutic twist. I haven't shown you, but we've done a lot of data to show that this enzyme system regulates the clearance of autophagic substrates like mutant huntingtin. So here's just one bit of data. In wild-type mouse embryonic fibroblasts, if one knocks down one of the kinases regulating the PI5P levels and thereby increases PI5P, you clear the mutant huntingtin more rapidly. If you overexpress the enzyme, you block the clearance and you accumulate the huntingtin aggregates. But in autophagy incompetent cells, these enzymes have no effect. So I believe this is a real new therapeutic target. It's potentially druggable, and we currently in the initial phases of a drug discovery exercise around these enzymes to try to identify novel inhibitors that are going to be useful. My lab's been very interested in, on the one hand, trying to find out how we can exploit autophagy to treat neurodegenerative diseases, but we've been also interested to see if autophagy is impaired in neurodegenerative diseases. And so there are multiple examples that have been discovered, and these are just a few studies from our lab, where we've shown, for instance, that excess alpha-synuclein blocks autophagy both in cell-based models and in vivo, that there's a mutation that gives you Lafora disease, which is a degenerative epilepsy that blocks autophagy by increasing mTOR activity, so it works opposite to rapamycin, and nitric oxide excess, as you see in Alzheimer's disease, would similarly block autophagy. And these all act at the level of autophagosome formation. There are mutations in the dynan machine which traffic the autophagosomes to where the lysosomes are in the cell. And we've shown that these mutations exist either in cells or in mouse models. They impair autophagosome degradation and autophagic flux and increase the levels of aggregates in the various model systems. And so I think that it's quite possible that dynan mutations or mutations in this machinery in forms of motor neuron disease or in Perry syndrome, which is a form of Parkinsonism, are impairing autophagy, and this thereby contributes to the accumulation of the aggregate-prone proteins and the toxicity. And finally, lysosomal storage diseases are the most common neurodegenerative diseases of childhood, and indeed, defects in the lysosome might be a very important contributor to forms of Parkinson's disease. And in collaboration with Andrea Balabio's lab, we showed that these lead to defective autophagosome or lysosome fusion. So now I just want to do a bit of a punt for basic cell biology, because I think in our lab it's shown us in the last 5 years how understanding the basic biology can really illuminate disease. So one of the key questions in the field is to understand how autophagosomes are built. And it's likely that there's membrane trafficking, and membrane come from multiple sources that contribute to autophagosome biogenesis. So the work we've done has shown that one key protein called ATG16 resides in a given set of clathrin-coated pits on the plasma membrane. and then traffics to the recycling endosome. And this trafficking event is critical for autophagosome formation. In addition, these vesicles undergo snare-dependent homotypic fusion events, which are also critical for the generation of autophagosomes. A distinct autophagy protein called ATG9 is found in separate clathrin-coated pits and is trafficked to recycling endosomes too, but through an independent route. This traffics through the classical route through early endosomes when it picks up a distinct set of snares to go to the recycling endosomes where it undergoes heterotypic fusion with the vesicles containing ATG9. And both the homotypic fusion and the heterotypic fusion events are critical for autophagosome biogenesis. This is physiologically regulated and we found that's increased in starvation conditions. And very recently, we've shown one mechanism how this might occur, and I'm happy to elaborate on it in the question time. So there's an actin regulating protein that regulates actin polymerization called annexin A2 that we found regulated the traffic of the ATG9 vesicles from the early endosome to the recycling endosome, and Morozova and colleagues showed about the same time that this regulates, in their hands, the endocytosis of the ATG16 containing vesicles and their homotypic fusion. So the annexin A2 protein works at distinct stages of this pathway I've described, and we've shown that it is upregulated through a JNK junk-mediated process after starvation, even in vivo in mouse brains. So why am I telling you all this stuff? So I'm going to give you two examples. There's a mutation in the retromer complex component called VPS35 that gives you an autosomal dominant form of Parkinson's disease. And in collaboration with Matt Siemens' lab, we showed that this particular mutation affects the ability of the early endosomes to recruit the actin remodeling wash complex and thereby affects the trafficking of the ATG9 containing vesicles to the recycling endosomes with resultant impaired heterotypic fusion. So this particular trafficking event is impaired as a consequence of this mutation, and we've shown that the consequent decreased autophagosome formation results in the accumulation of alpha-synuclein in cell models with this mutation. The second example comes from a large collaborative study driven by Kevin and Moreau in my lab and Angie Fleming in my group, who works on zebrafish models. And what we showed in this study is that this CALM locus, which is a well-validated Alzheimer's disease risk locus, affects a number of stages of the autophagy pathway. This protein affects the endocytosis of key snare proteins, and it affects the endocytosis of VAMP2, which impacts on the homotypic fusion of ATG16 molecules, VAMP3, which impacts the heterotypic fusion of ATG9 and ATG16 molecules, and these two affect the formation of autophagosomes. It also affects the endocytosis of VAMP8, which affects the fusion of autophagosomes and lysosomes. So it affects both autophagosome formation and autophagosome lysosome fusion. And we've gone on to show in neurons, in Drosophila, and in zebrafish that affecting this pathway through CALM, which is also known as PICALM, affects tau toxicity and tau accumulation. So it provides a link. It's almost certainly not the only link, but a link between a GWAS hit, autophagy, and tau accumulation as a hallmark feature of Alzheimer's disease. So to end the sort of the major part of the talk, I want you to take home two messages. The first is that autophagy upregulation might be a viable therapeutic strategy for suitable neurodegenerative diseases by enhancing the clearance of these toxic aggregate-prone proteins, by protecting against apoptotic insults, and this can be mediated through a range of targeting, a range of signaling pathways. On the other hand, autophagy compromise might be an important pathogenic feature driving disease in certain conditions. The final story that I'll briefly tell you about has got nothing to do with autophagy, and it's a recent story, but we've been working for many years, which has been driven by Maria Jimenez Sanchez in my lab. She's Spanish, and it's the fruits of a really successful and fun collaboration we had with CIIIEN Biotech. And the story started with a genetic screen. So we developed cell lines that expressed full-length mutant huntingtin, and these cells show some toxicity. And we then exposed the cells to what we call a druggable genome screen. So it's the genes in the genome that are potentially druggable, about 6,000 genes, and we knocked down each one in turn with siRNAs and did all the standard deconvolution type experiments one would do to try to ensure the effects were not due to off-target effects. And at the end of the day, we had about 200 genes that were validated targets showing an effect in the cell-based system. I didn't really believe the results, so I thought we needed to do a completely orthogonal validation. So we took a Drosophila model that we had in the lab that expresses mutants, in fact, just a fragment of huntingtin, in fact, just the polyglutamine expansion, and we found the Drosophila orthologs of the hits. There are about 130 Drosophila orthologs we identified, and then we tested them with knockdown experiments in Drosophila. So if you express expanded polyglutamines in the Drosophila eye, you get these necrotic spots, and these are rescued by about half of these genes, which I thought was a pretty good hit rate. For the rest of the talk, I'm just going to focus on one of the hits called QPCT. I'll explain what QPCT is in the next slide. But we show that QPCT rescues like the other genes, and QPCT also decreases the number of aggregates formed by GFP-tagged polyglutamine expansions in the eye. So this shows that the aggregates formed by the GFP tag protein, and you can see they decrease by the QPCT knockdown. This also occurs, HEX cells expressing exon 1 of mutant huntingtin, as well as in primary neurons. So this shows the aggregation data with the knockdown, and likewise in the primary neurons. So what is this enzyme? QPCT is called glutaminyl cyclase, and it converts an N-terminal glutamine or glutamate to a pyroglutamate. And this has received some attention in the context of Alzheimer's disease, where removal of the N-terminus of the A-beta peptide reveals a glutamate, which can then be pyroglutamated by this enzyme, and it's thought that this increases its hydrophobicity and aggregation potential. And so we knew that it was possible to develop drugs against this enzyme. And so CIIIEN Biotech did an in silico screen to identify molecules that could potentially inhibit this enzyme. These were then tested in a biochemical assay, and then we tested the 3 hits that came through the system in cell-based and in vivo assays, and found that the drugs that inhibited this enzyme were protected in cells, primary neurons, Drosophila, and zebrafish. So this shows some of the data, for instance, in the cells, on the neurons, you get protection with these 3 drugs, and the effects against aggregation, for instance, are dependent on the enzyme. So if you knock down the enzyme, there's no further protection with the drugs as opposed to the cells treated with the control shRNA. I don't know if you can see, here are some of our zebrafish data showing what happens when you treat the zebrafish model of Huntington's disease with this drug. So we generated zebrafish model of Huntington's disease that expresses exon 1 of Huntington in the rod photoreceptors. So this is the retina of the zebrafish, and you can see very clear aggregate formation by the GFP tag protein. And you can see the drug reduces the number of aggregates that are formed, and I'm sure you can't see it. But here we've stained the retina for rhodopsin. So the mutant huntingtin's only expressed in the rods, so you can look at the number of remaining rods by doing a rhodopsin stain, and the rhodopsin staining is much improved by the drugs compared to the DMSO condition. So how does the QPCT modulate aggregation? It depends on its activity, because if you overexpress QPCT, you get more aggregation. If you express inactive form of the enzyme, you have no effect. It affects many different aggregate-prone proteins, so it affects GFP-tagged Q81, so it's a polyglutamine expansion, but it also affects a polyalanine expansion. And it appears to affect the oligomerization at the early stage of these proteins. So here's an experiment where Maria's overexpressed GFP-tagged mutant huntingtin fragments and flag-tagged mutant huntingtin fragments and immunoprecipitated. And if you treat with the drug, you get less immunoprecipitation, so less interaction of the soluble interacting protein. How does this work? It doesn't affect autophagy, it doesn't affect the proteasome, it doesn't affect the integrated stress response, it doesn't affect HSF activity, it doesn't affect heat shock protein 70. We tried everything. We got desperate. When you get desperate, you do an array. And so we exposed a stress array, cells to this drug, and looked at expression of proteins on a stress array. And the best hit was the chaperone called alpha-beta crystalline, which we were excited about because we knew that overexpression of this protein in other contexts could alleviate aggregation. This is confirmation with the knockdown of the protein of QPCT in primary neurons, showing that the levels of the alpha-beta crystalline go up. And here the protein data showing that the levels of the alpha-beta crystalline go up either in naive cells or in cells expressing mutant Huntington when you knock down the QPCT. The effects of the QPCT on the aggregation look like they're largely dependent on the alpha-beta crystalline, because this again is in naive cells where the drug decreases the aggregation. But if you overexpress alpha-beta crystalline, you reduce aggregation, but the drug has no further effect. So we left with the pathway where we have a druggable screen, genetics, we identify a drug, we identify the mediator, the primary mediator of the drug, and this looks like that QPCT is reducing the aggregation not only of mutant Huntington, but of a range of other proteins by modulating the levels of the small heat shock protein, alpha-beta crystalline. So I'd like to leave you on a more optimistic note. I think there are likely to be multiple protective routes that we can consider for these types of diseases. I've talked about two, autophagy and small heat shock protein chaperones. We certainly need to consider the possibility of synergy and interplay between these pathways, and these are areas we'll be looking at soon. Finally, this is work done by many people over many years. I've mentioned the key players as I've gone along, but I just want to re-acknowledge some major collaborators we've had over the years. So Steve Brown in Harwell helped us with the motor neuron disease studies in mice and with some of the alpha-synuclein studies in mice. Kihiro Kane has been our long-standing partner in crime doing Drosophila studies of these diseases. Andres Flota, I've mentioned the context of the tuberculosis work with Diane Ordway. Olaf recent tubing and very kindly gave us the spinocerebellar ataxia type 3 mice before they were published. Roger Barker's in Cambridge has been collaborating us with the clinical studies. Paul Cudden made the zebrafish model in our group. And Tristan Renaud with the oyster study I mentioned. Andrea Caricasole was the main player to Sierra Biotech in the study I've just mentioned on the QPCT, and Andrea Balabio with the study on the lysosomal storage diseases. And finally, I couldn't have done the work without the generous funding we've had over many years. Thank you very much.
Okay, questions for this lecture? Okay, there.
Could this mechanism mediated by glutaminyl cyclase similar to the role proposed for transglutaminase in protein aggregation and apoptosis?
We haven't formally tested that. Actually, in the old days, we did do some work with transglutaminase. The transglutaminase effect is thought to be more direct in that it's supposed to be working catalytically on mutant Huntington. And certainly in vitro, there's some support for that. The question is, does transglutaminase have other substrates? Is it playing into this pathway? We haven't looked. It's something that we can look at. It's, but we hadn't thought to, but maybe we'll do that. Thank you for the idea.
Other substrate. Thank you.
Yeah. I mean, I think the point is we have quite a lot of data suggesting the QPCT is not working directly on Huntington. It's working via the QPCT, and that was one reason to think against what you suggest. But it's quite plausible that it could be acting through, it might be another way of regulating the expression of the QPCT.
Okay, I have some questions. Perhaps some are very obvious and you can answer very easily. You talk also about that the ratio of two lipids, PI3P and PI5P, is important for autophagy. My question is any knowledge about the levels of these lipids in diseases related with autophagy, like Huntington disease or...
Okay, that's a really good question. I'm going to duck it and then tell you why I duck it. So there is a literature suggesting that there's impaired activity of a protein called Beclin-1 in Huntington's disease and Alzheimer's disease. There's a literature, I'm not saying it's right. And Beclin-1 is a key protein that regulates the site of activity of VPS34 in the context of autophagy. So it's quite plausible that the PI3P mechanism is impaired in Alzheimer's disease and Huntington's disease, and possibly in some other conditions. The reason I'm a little bit cautious about answering your question in a direct way is that for these lipids, what matters is not only how much there is in the cell, but also that it's doing the right thing at the right place at the right time. So it's a dynamic and location-dependent phenomenon, and so I think it's quite difficult to say for certain. But there are inferences with relation to your first question, and indeed, that might provide further support for using the PI5P as a bypass strategy. Thank you.
Other questions?
Regarding to the same PI5P, and whenever you inhibit the production of PIP2, then you are as well provoking an imbalance on this lipid that is really important for any signaling, well, many of them. So I'm sure that if you add PI5P, sorry, you are getting a benefit, but will you affect as well the balance of the other one that is really important for the cell? Thank you.
So actually, before we did the PI5P work, we did a lot of work on PI45P2. The levels of PI45P2 are much, much, much higher than PI5P. So if we perturb PI5P metabolism, the effects on PI45P2 are negligible. So I think that probably answers your question, yeah. It's a very, very low abundance lipid.
Okay, I have also a short question. It's about, you know that there's a role of microtubules in autophagy. And also, recently there is also some funny aggregate of microtubules, tau, in Huntington. Have you tried to look for some targets related to microtubules or to the stability of microtubules in this kind of diseases?
Yes. So we've done quite a lot of work, which is unpublished on microtubules. So some of the published work is as follows. The published work we've done, and I think is confirmed by others, is that microtubules are critical for getting the autophagosomes to the autolysosomes. However, there's a distinct role. So Zevi Elazar from Israel published a paper which we initially were a little bit skeptical about, and he said microtubules are important for autophagosome formation. And he's right. The microtubules are important both for the delivery of the autophagosomes to the lysosomes and for a number of steps probably relating to autophagosome formation. And we've been chasing that a little bit in the lab. So I think it is complicated. In the context of tau, the little bit of playing we have done with tau does not reveal an obvious effect of tau on autophagy, as far as we can see. We've tried doing some experiments. I'm not saying they are exhaustive or complete, but we had a bash at it. So whether normal perturbations one would do with tau are sufficient to disrupt these steps, I don't think so. I mean, you might have to push it very hard.
Okay, another question? If it's not another question, I think that we will have a coffee break and we will be here at 5. At 5, not 5:10, 5. Okay, see you.